Hello students, today this video is about history of enzymes. In fact, much of the history of biochemistry is about the history of enzyme research. The history of enzymes may be regarded as commencing with the work of Dubrenfort, who in 1830 prepared malt extract from the germinating barley seeds. This extract possessed the power of converting starch into sugar. Another important discovery, which is often referred to as the dawn of biochemistry, dawn, D-A-W-N, dawn of biochemistry, was the discovery of the first enzyme, diastase. Diastase is also called as amylase by Payne and Persos in 1833. Actually, before that, the role of saliva in converting starch to sugar and the stomach secretions in digesting the meat were known. Even the conversion of starch to sugar by plant extracts also was known. But the factors responsible for these conversions were unknown. So, Payne's discovery provided the answer. Then, in 1850s, Louis Pasteur, the French chemist turned microbiologist, who is also called as the father of microbiology, he believed that fermentation was catalyzed by a vital force present in the yeast cells, and he called this vital force as ferments. These ferments, he believed, could function only inside the living organism. So remember, he gave this vitalistic theory that the ferments or the enzymes, which we now call them as, they work under living conditions only. Then in 1878, Cohne coined the term enzyme. Then by the end of the 19th century, Edward Buchner, a German biochemist, in 1897 for the first time demonstrated fermentation using yeast extract. This was the first evidence to support that biochemical processes could occur outside the cell even without the presence of living cells. Now, this discovery completely discarded the vitalistic theory of Louis Pasteur and convincingly demonstrated that the fermenting principle or the fermenting substance could be present even outside the cell. So Buchner named this enzyme as zymase. He was awarded Nobel Prize in Chemistry in 1907 for his discovery of cell-free fermentation. You know, students, he was a major in the German army and he died in action on the Romanian front in 1917 during First World War. So this discovery by Buchner was important in the history of biochemistry because it opened way for the extraction of enzymes and also the in vitro studies of the enzyme. Then another important breakthrough came from Sumner. He was working at Cornell University and he isolated and crystallized the enzyme urease. He isolated it from jack bean, also called as Cannevalia ensiformis, and he found that the enzyme urease crystal was entirely protein. So this suggested that all the enzymes, they were protein. After Sumner, when he was young, he lost his left arm as a result of hunting at the age of 17. So he was discouraged by his teachers from pursuing a career in chemistry. They felt that he was too handicapped for the profession, but he belied all these. He is called as father of modern enzymology. He received the 1996 Nobel Prize in Chemistry for crystallizing the first enzyme along with his compatriots Northrop and Stanley for his work on preparation of enzymes and virus proteins in pure form. Then in 1930 till 33, Northrop isolated and crystallized pepsin and trypsin, and he also confirmed that yes, they are proteinaceous in nature. Then in 1967, Woos, Crick and Leslie Orville first suggested that RNA could also work as catalyst, but this was a mere suggestion. It was only in 1980 when Thomas Seck, who was studying RNA splicing in tetrahymena thermophila, and Sidney Altman, who was working on the bacterial RNase P complex, first uncovered the catalytic property of RNA. Thus, ribozymes were discovered. Then, after 14 years, Breaker in 1994 reported the catalytic property of DNA also. So such catalytic DNAs, they are referred to as deoxyribozymes. But it was demonstrated only under in vitro condition. So the discovery of enzymatic nucleic acid was a big blow to Sumner's postulate that all enzymes were proteins, but it certainly opened new dimensions in enzyme research. So coming back to the part which I discussed in the first video regarding the definition of enzymes, that yes, enzymes are biocatalysts which alter the rate of specific chemical reactions, but we also added that with the exception of few catalytic RNAs, all known enzymes are proteins. So by studying the history of enzymology, you can easily understand that yes, with the exception of a few catalytic RNAs, all known enzymes are proteins. Why we have not included the DNAs? Because the deoxyribozymes have also been characterized having catalytic property, but they have not been discovered under in vivo conditions. Thank you.